Hello and welcome. Today we are going to discuss the first chapter of class 11 zoology, and that's animal kingdom. Well, I hope that you have gone through the five kingdom classification in your previous classes. For example, let me show it here. Yes, this is the five kingdom classification, and this was given by Robert Hardinge Whitaker. So, in this classification, he divided all the organisms, whether they are in air, on the surface of the earth, or in water. He divided all those organisms, the entire set of organisms, into the five kingdoms. And the five kingdoms are Monera, Protista, plants, animals, and fungi. So these are the five kingdoms. Okay. Why he divided them? Because it would be convenient for us to study them in detail, and we will get a lot of information about them, so that we can know about the surroundings which we are covered with. So this was the five kingdom classification, and it was given by Robert H. Whitaker, and this is the most advanced classification, and it's universally accepted by all over the world. Now, out of the five kingdoms, the kingdom which we have to discuss here is the animal kingdom. Now, the animal kingdom, as you know, it shows a great diversity of life in comparison with the plant kingdom. So I can write it here as that it shows great diversity. Well, when I say diversity, it means you can see a number of animals, okay, number of animals, a number of species living within the surface of the earth, or underwater, or on the surface of the earth, and even on trees, and even in the air. So this is the diversity. Okay, so that's why in nutshell we can say the animal kingdom shows a great diversity as compared to the plant kingdom. Okay, all animals. When I say all animals, it means all those animals which either live in, which are either aquatic, which are terrestrial, which are aerial. So all animals, they possess three main characteristics. Okay, they are multicellular. Okay, they are eukaryotic. It means they have advanced cell type, and they possess heterotrophic mode of nutrition. Heterotrophic mode of nutrition. So, all animals, they possess these three main characteristics, and that's why they are called metazoans. So we can say all animals, they are also called as metazoans. Okay, they are called metazoans. Now, the metazoans, they have, I will write down it here. Metazoans, they have been divided into two subkingdoms. Okay, subkingdoms. One's called the parazoa, and another is called the eumetazoa. One is called a parazoa, and another is called eumetazoa. Okay, or we can say in other words that it has two types: parazoa and metazoa, eumetazoa. Well, the parazoa includes sponges and eumetazoa. Eumetazoa include rest of the animals. Rest of the animals. Okay, let me clear this thing here. Okay, so Eumetazoa may include the rest of the animals, okay, other than sponges, because sponges have been included in a separate subkingdom, and that is Parazoa. Okay, now why they have been kept in a separate subkingdom? It's because sponges have a separate, or we can say the different kind of a body plan, and in sponges the cells are loosely aggregated. Okay, they do not form tissues. They do not form tissues. So here this cross means it represents do not. Whenever you see this cross in my lecture, do understand that this means do not. So they do not form tissues. Sponges do not form tissues. Okay. Now coming to the Eumetazoa. In case of Eumetazoa, the cells get structurally and functionally modified and they form tissues. So here you can see tissues, not just tissues. Also tissues get differentiated into organs, and these organs may get combined and they form systems, and they are known as organ systems. Okay, so this was the introduction of the animal kingdom. So you have to keep 2-3 things in mind in this introduction, that all the animals are multicellular, eukaryotic, and heterotrophic, and that's why they are also called as metazoans. While the metazoans have been divided into the 2 subkingdoms, one is called the Parazoa and another is called the Eumetazoa. And in Parazoa we include sponges, and in Eumetazoa we include rest of the animals. Okay, it's Eumetazoa. E here remains silent. Okay, now the Parazoa, it consists of the sponges, and sponges are nothing but their body plan is different. That's why they have been kept in a separate subkingdom, and that's called the Parazoa. And here the cells are loosely aggregated. They do not form tissues. Well, in opposite to the sponges, here the rest of the animals, they form tissues and they form organs, they form organ systems. Okay, so this is about the animal kingdom introduction. Now we have to study, let me choose another page here. We have to study what is the basis of classification. Basis of classification. Okay, so what's the basis of the classification? This is important here. What are the characters which have been used in order to classify the animal kingdom? Okay, now now let's study them one by one. The first character that has been used in order to classify the animals is here. Let me choose another color. Is the labels of organization. Labels of organization. This is the first character that has been used to classify the animal kingdom, so that it would become easy for us to understand the animals, the wide range of the animals, the diverse kind of the animals and their species, to study them one by one and in detail. Okay, now this following are actually the major types of the labels of the organization. though all the members of the animalia are multicellular, all of them do not exhibit the same pattern of organization of cells. For example, let's take the first, that is cellular level of organization. Cellular level of organization. Now, in cellular level of organization, you will see that the cells are arranged as loose aggregates. Here, cells are loose aggregates. Okay, they form loose aggregates. They are not compactly packed, just like in humans and other animals. So cells are loosely aggregated. Okay, just like this. They're not compactly packed, just like in we humans. We possess that character. So in cellular level of organization, cells are loosely aggregated. Okay, and number second, there is no coordination between cells. So there is no coordination between cells. Okay, they do not work in coordination. They don't show continuity in their function. Okay, so but individual cells they are capable of performing specific functions. Individual cells, not a group of cells. If a group of cells will perform a particular function, then it means then there is a tissue level of organization. So, but here the individual cells, we say this cell performs a different function, this cell performs different function, this cell performs different function, and that's why all the cells, or for example, a group of cells do not form a particular function or do not carry out a particular function. That's why here they cannot be called a tissue. Okay, hope you understand it. The best example for this cellular level of organization can be seen in sponges. That's why in my previous lecture I told you that here you can see that they do not form tissues. That's why I said this. Okay, coming back to our topic. So the cellular level of organization can be seen in case of sponges. Okay, we have to discuss them in detail, but here you have to keep this in mind. So this is the cellular level of organization. Okay, coming to the next here level of organization. Now that's called as tissue level of organization. So I will write down it here as tissue. Keep in mind, when we say tissue level of organization, it means there is a formation of tissue. Okay, tissues are formed here. So let us discuss them. Actually, the cells performing similar function are aggregated to form tissues. Cells, a group of cells which perform particular function or which perform similar function. Okay, they form tissues. Okay, here keep in mind group of cells. So I will write down here as group of cells performing same or similar function. They form a tissue. Okay, that's called the tissue. So the tissue level of organization, where we can see them, the tissue level of organization can be seen in coelenterates. Okay, in coelenterates we can see the tissue level of organization. Tissue level of organization. The best example for the coelenterata this phylum has is Hydra. Okay, is Hydra. So this is the tissue level of organization. So in tissue level of organization, we can see that those organisms which have tissues, okay, their highest level of organization as tissue, then it means that they possess tissue level of organization because their cells a group of cells, they get combined and they form a particular or similar or a particular function, and they form tissues. That's why that organism or that phylum or that group possesses tissue level of organization. And the best example for the tissue level of organization is exhibited by coelonterates, also called as cnidarians. We will discuss them in detail. These groups, we have discussed them in detail, but keep in mind this time. Okay, up comes another here, the level of organization, and that's here organ level of organization. Organ level of organization. Well, tissues get grouped together and they form organs. And when they form organs, it means that the organ level of organization comes into the existence. Okay, so each organ carries out a specific function. Each organ carries out a specific function. Specific function, just like stomach is the organ in we humans. Okay, just like we have a liver, just like we have here small intestine. So these are the organs, just like pancreas. But they do not form, in case of organ level of organization, they don't form systems. Organ level of organization, in case of, they don't form systems. So we can say any organism that possesses organs, they are known as organ level of organization. Okay, let me give you an example here. The best example is platyhelminths. And other higher phyla also possess this level of organization. Okay, and other higher phyla. Not just phyla, you can choose here to say phyla, because phyla is the plural term for phylum. Okay, other higher phyla. So here we understood the three levels of the organization: that's cellular, tissue, and organ. Okay, now we have another level of organization here, and that's called as organ system level of organization. Hope you understood what we studied or what we discussed. Now the organ system level of organization, in this, a group of organs, I will write down it here, a group of organs, okay, working together. Group of organs start working together and they form a system called as organ system. Okay, let me give an example. I will give you the best example. For example, we have here digestive system. So we say digestive system, so it means it consists of many organs that work to perform digestion. Okay, for example, in digestive system we include organs like we have mouth, we have teeth. Teeth also help in the digestive process, okay, because it helps in the churning and the breaking down the food. Okay, we have here salivary glands. I will simply write them as SG, salivary glands. We have pharynx. We have esophagus. We have stomach. And we have small intestines, SI. large intestines are LI. Okay, and we have other glands also. So all these organs, they combine together and they perform a particular function, and that's called the and they form a system that's called the digestive system, and the function performed by them is the digestion. So that's why these organs get combined together and they carry out the process of digestion. That's they carry out digestion. Well, we have here, for example, in some, this is important point for you. We have here, for example, incomplete system and complete system. In complete system. For example, in case of incomplete system, you will find out that the digestive system has only one, or we can say the single opening. Okay, there is only single opening, and that single opening is that acts both as mouth and ends. This single opening acts as mouth at the time of ingestion, when the food is taken in, and acts as ends at the time of egestion, when the undigested food is expelled out. So this system, where there is only one opening or a single opening, and that single opening acts as both mouth and ends for the ingestion and the egestion processes respectively, then that system is called as incomplete system. Or we can say, if we are talking about the digestive system, it means the incomplete digestive system. Now, talking about the complete digestive system, here two openings are present, and one opening is called a mouth and another is called as ends. Well, this opening is used for the process of ingestion, and this is used for the process of egestion. Okay, so this means that this is the complete digestive system. Well, these were the levels of the organization, and we studied cellular level of organization, and after that the tissue level of organization, after that comes organ level, and that comes after organ system level of organization. So we discussed all these four levels of the organization. So this forms the first character which we used for the classifying the animal kingdom. For classifying animal kingdom. Okay. Now, coming to the another system we have, for example, here, which has been given in your textbook, is the circulatory system. Okay, circulatory system. Well, as you know, the circulatory system deals with actually heart, arteries, veins, capillaries, blood, plasma, etc. Okay, so here you can see that the organs like heart, arteries, veins, capillaries, these all organs, they combine together and they form a system, and that's called the circulatory system. Okay, and this circulatory system is also of two kinds, is also of two types. One is called the open type, and the another is called the closed type. That is discussed there. Now, in open type, you will see that blood is pumped out of the heart. Blood is pumped out of the heart, and the cells and tissues are directly bathed into it. Directly bathed into it. So it means that there are, when remember or recall when I told you that the cross represents here do not. It means that arteries, capillaries, and veins, they do not play any role here. It means they are present. They are absent, sorry. Okay. Now, so in open type, the blood is pumped out of the heart and the tissues and the cells. Okay, they get directly blood from the heart, we can say, and or we can say they are directly bathed into it, and there are no arteries, no veins, and no capillaries. They are totally absent here. Keep in mind, in open type, the arteries, veins, capillaries, so we can say the blood vessels are not present. They don't have any role here. Now, talking about the closed type. Now, in case of closed type, so first of all, let me give the example for this open type. Open type has the best example as the arthropods, and molluscus. And talking about the closed type, in case of closed type, blood flows inside the blood vessels. Inside the blood vessels of varying diameters. When I say varying diameters, on the basis of the diameters, the blood vessels are of three types. I will write down them here. Arteries, veins, capillaries. Okay. The best example for the closed type is man. We can say the human. So this is the best example. We have blood vessels, we have arteries, we have veins, we have capillaries that carry blood from one part of the body to the another part, from the heart to the far-flung organs of the body, to the stomach, to the liver, and to other parts of the body, urinary bladder, and many other parts. So through these blood vessels, so we have closed type circulatory system. Okay, so this was the first character that we discussed here, and that is the levels of the organization. So on the basis of the levels of the organization, animal kingdom has been divided, okay, into many subkingdoms or groups or phyla, okay, or the phyla. So now let's discuss our second main character, and that is symmetry. Now the symmetry. Symmetry. This is the second important character that has been used for classifying the animal kingdom. Okay. Now, first of all, let me make you understand what actually is a symmetry. Symmetry means, what's the definition of the symmetry? Symmetry means, symmetry actually means the arrangement of the body parts. Arrangement of the body parts body parts, okay, on the sides of the man, on the sides of the man, median axes of the body. Of the body. And the important point here is this idea was actually given by Haeckel. This idea was actually given by Haeckel. He was a great biologist, and the idea of symmetry was given by him. Okay. Now let's take the example. Through the example, you will understand the fact. So look at these two figures here. Okay, this is just like a tree here. And look at these two figures. Here, if you see that, if I will cut this figure, sorry, if I will cut this figure as, I will name this figure as A and this is figure B. So I will cut this, if I cut this figure through this median line, its right part will be identical to this left part. Okay, and take the example of figure B. If I will cut this figure through this median line, do you think its right part will be equal to its left part, identical to its left part? No, this will not be. Because if you see here, the here structure is different, and at the same time, if you see here, the structure is different. So this part will not be identical to this part. And this, and in this figure A, both the parts or both the halves will be equal, just like a mirror images of each other. But in case of figure B, you will see that they don't form no mirror images. Both the halves are not identical to each other, or we can say the mirror images of each other. So we say that part A or figure A, this shows symmetry, and the figure B, this doesn't show any kind of symmetry. So part A, figure A is symmetrical and figure B is asymmetrical. Hope you understand through this example. Now, getting back to the topic. Now here, in case of animals, there are three types of symmetry. There are three types of the symmetry. Number first is asymmetry. The first is asymmetry. The first is asymmetry. So I just told you through this example that here you can see the figure B, that this figure B is asymmetrical, meaning if we cut through its median or its main axis, you will see the halves are not identical or the mirror images of each other. So we can say this is asymmetrical. Now same is here. I'll write down its definition as body cannot be divided into two equal halves. Okay, throw any plane passing through the center. Throw any plane passing through the center. Okay, center. So this is called the asymmetry. There are many examples to the asymmetry, just like you have your hand, which is also asymmetrical. Okay, hope you understand the asymmetry. Now, our second point here, second type. This is the first type. Now coming to the second type, which is called radial symmetry. Symmetry. Please be patient so that you can understand the topic easily and you will never forget. In radial symmetry, the body can be cut into two exact halves. Cut into two exact halves. Exact halves through any plane. Through any plane passing through central axes. Okay. Such animals have two ends in the main body axes, the oral and aboral end. Two ends. Oral, second, let's start. Oral and aboral end. It's got two ends, oral and aboral ends. But no left and right sides. Here, in case of radial symmetry, you will find out that there are the two ends. One's called the oral end and another is called the aboral end. But there is no left and the right end. By example, I am going to show you here so that you can understand it. But before that, let me give an example for this because so that it will be easy for you to understand. Well, the example for I forgot to give you the example to the to the asymmetry. So the best example for the asymmetry is sponges. Okay, sponges do not possess any kind of the symmetry. Okay, it means if we cut the sponges into any plane, okay, in any plane, uh, its halves will not be exact copy to each other, or they will not be equal to each other. They will not be identical to each other. Okay, so that's why they are just like we have hand. Our hand is asymmetrical, so it means that they are also the sponges are also asymmetrical. Okay, and here the best example for the radial symmetry is cylantrates, also called as nitrogens. Okay, and another group here, ctenophora or ctenophores, and echinodermic. Okay, okay. Now let's see the example. What's up? Here you will see the radial symmetry. This is a radial symmetry, and this the organism here. This is actually the height, and you can see if you will cut this height through any plane like this. Here, this is the first plane, and I cut this first plane. So it is this hollow will be identical or equal and exact copy of this hollow. Okay, and similarly, if we cut through this plane, this hollow will be identical or equal and exact the mirror image of this hollow. Okay. Similarly, talking about this, sorry, this this uh, this cutting, this cut will make, or this plane will make uh, that portion identical to this very particular portion. So these are actually the planes of the symmetry, and the organism here is from the Cylantrate of phylum, and this is Hydra. Okay. Now, getting back to the topic, we are going to learn the third particular type of symmetry, and that is that is that is bilateral symmetry. bilateral symmetry. Okay, here we have bilateral symmetry. In case of bilateral symmetry, you will find that the body can be divided into two identical halves. Identical halves only through a single plane. Only through a single plane. Okay, only single plane can be through the single plane. The two identical halves we will get from a body. Okay, and passing through the central axis of the body. Okay, okay. Well, these animals have front and rear end. Okay. Similarly, they can also have upper and lower side, upper and lower side, and they can also possess equivalent left and right side. Hope you understand. But to make it much more uh, of this concept clear, to make this concept much more clear, let's take an example here. Best example for the bilateral symmetry. Best example for the bilateral symmetry here is analysis, arthropods, okay. Let's take the example. Let's see the diagram. Let's see the image of an animal. Here we have a lobster. Okay, you will see that only one plane has been given. Okay, only one plane has been given. As compared to the hydra, or in case of the radial symmetry, you will see that different kinds of the planes have been given. The planes of the symmetry, but here is only one plane. And if we will cut this lobster through this plane, through the median of this body, through the median of this body, through the median of this body, you will see that this part, this left, we can say, for example, let's consider this left portion of this body will be identical to the right portion of the body, or right part of the body. This is this anterior end, this is this anterior end, and this is posterior. Okay. And this is the dorsal side, this upper side, okay, it's back side, we can say, and this is the side. This is the plane symmetry. Okay. And this is representing the bilateral symmetry. So I hope you understand through this example what's the difference between the radial symmetry and what is bilateral symmetry. Only single plane is here, and here are many planes. Okay, and any plane. Okay, here is left side, right side, lower side, upper side, okay, and front and rear end, okay. But here you will see nothing like that. Okay, so this is the radial symmetry and this is the bilateral symmetry. Hope you understood the symmetry, which is the second most important basic character which has been used to classify the animals. Okay. Now, let us discuss the geometries. Now, this classification is based on the number of embryonic cell layers, or we can say the germ layers. Embryonic cell layers are also called as germ layers, and they can be divided into two. Okay, so here we have two on the basis of the germ layers. How many germ layers are present in an animal? We have divided the animal kingdom on the basis of this classification into two groups. One's called the diploblastic animals, and another group is called as the triploblastic animals. Well, in case of first of all, let me tell you the three germ layers are the first is ectoderm, and number second is the mesoderm. This lies in between. And number third is and well, this is outer layer, and this is middle layer, and this is inner. Okay. The diploblastic animals, as the name here signifies, di, it means two, and here the name signifies tri, it means tri, which means three. So diploblastic animals are those animals which possess two embryonic cell layers. Okay, which possess two embryonic cell layers, or we can say the geometries. That is outer ectoderm and inner endoderm. Okay, as the name signifies, ecto, which means outer, and the endo, which means inner. So there are the two layers present in the diploblastic animals. One is called the outer ectoderm layer, and another is called the inner endoderm layer. Okay. Well, in between the ectoderm and the endoderm, there is undifferentiated mesoglia. Okay, which is not differentiated in the mesoderm. That is why this cannot be considered as the mesoderm. That's why it's undifferentiated. And well, the diploblastic animals include coelotrates and sponges. Okay, coelotrates and the sponges are the best example of the diploblastic animals. Now, talking about the triploblastic animals, well, as the name signifies, they contain three embryonic layers, or we can say that germ layers. That is outer ectoderm, middle mesoderm, and inner endoderm. Okay, and the example for this group of animals include platyhelminths. Okay. Don't worry, we will discuss these phyla just like we have coelenterate, sponges, platyhelminths, estelminths. So we are going to discuss them one by one in detail ahead. So don't be worried. Well, these triploblastic animals are actually those animals which possess the three embryonic layers of the germs. Well, all the tissues, why they are so important? Because all the tissues and organs are formed from these layers. Just like, I will take example here, ectoderm, we have ectoderm. Ectoderm gives rise to the epidermis, just like we have second. So it gives rise to the epidermis and nervous system. Ectoderm gives rise to the epidermis and the nervous system. Similarly, mesoderm gives rise to the muscles. Keep in mind, this is an important point for you. This has been asked in many exams. Endoskeleton, reproductive organs, and most important, coelom that we are going to discuss ahead. Okay, and the inner layer, that's called the endoderm. The endoderm gives rise to the alimentary canal, that's AC. Don't get confused with AC, that is an alternating current, but it's actually the alimentary canal. Well, lungs and many other organs, etc. Okay, so these three layers, they are actually the embryonic cell layers. They are very important, and their formation is called as gastrulation, which is an important process at the time of the embryo development. Well, that is a separate topic. Well, these three layers can be understood by taking an example on a diagram. Let's take a diagram here. So here you can see this outer layer. This is called this purple color layer. This is actually the ectoderm. Here you can also see this is ectoderm. And in between this a reddish color. Well, you can see that this is simply the reddish color, and they are just like in the ectoderm. The ectoderm possesses these cell-like structures, but in this reddish color, there are no cell-like structures because this is undifferentiated. This is not differentiated as mesoderm into mesoderm. That's why this is undifferentiated. And the innermost layer here, this is called the endoderm. Well, this inner layer has been differentiated into endoderm. That's why the animals which possess this kind of we can say the pattern. It means they are known as diploblastic animals because there are two well differentiated layers. That's ectoderm and endoderm. Well, the mesoderm here is not. We cannot call it as mesoderm because it's actually the mesoglea, which is an undifferentiated mass. Okay, so this is the example of this is the pattern of the diploblastic animals. And talking about this pattern, okay, here you can see the differentiated ectoderm. Okay, here you can see the differentiated mesoderm and the inner layer. This is the endoderm. Well, this and all the cells, even in this middle layer, you can see the differentiated cell structures can be easily seen. So it means it's completely differentiated into the mesoderm. That is why it means it consists of 3 layers, and this consists of only 2 layers, 2 differentiated layers. Okay, and this pattern can be seen in triploblastic animals. Well, the white cavity here and here, this is actually the digestive cavity. Okay, so let's get to the topic here. I hope you understood this. Okay, so this was the third important character that we discussed, which has been used for the classification of the animal. Now, another important character that we have to discuss in the classification of the kingdom is the body plan. So let us discuss the body plan. Body plan. Body plan. Well, this is a body plan. Okay. This is a body plan. Well, talking about the body plan, the different animals can have different pattern, or we can say the setup of the body plan. Just like, for example, in case of sponges, you will be able to see that cells are loosely aggregated. Okay, cells are loosely aggregated. So it means that we can see there is cell egg type of body plan in case of sponges. Okay, and in case of coelenterates and platyhelminths, you will be able to see the blind sac type of body plan. And here in this, the alimentary canal has a single opening. Okay? The alimentary has got a single opening, and that single opening serves as both mouth and anus. Okay, mouth and anus, both. So this is called the blind sac type body plan. And same is the case with the and the third, same is the case with the coelenterates. And the example here is also the platyhelminths. Well, the third type of body plan here is tube within tube. Here it's actually tube within tube. And this type of body plan can be found in platyhelminths, annelids, arthropoda, mollusca, and coelenterates. Okay. Well, in this body plan, the alimentary canal, we can see also the digestive system is continuous, having anus, having opening at each end. You can see the openings at each end. At the one end, you can see the mouth. That's called the mouth, used for ingestion, and at the other end, you can see the anus used for egestion. Okay, so there are 2 ends. Well, on the basis of the evolutionary lines, the organisms can be protostomic and deuterostomic. Well, talking about the protostomic, the protostomic animals are those animals in which mouth are those animals in which mouth is developed, mouth gets developed before ants. Okay, mouth is developed before ants. Okay, it means I mean to say that ants develops later. First mouth develops. Okay, and this can be seen in the protostomic animal. This example of this kind of evolutionary line can be seen in annelids, mollusca, okay, molluscus or mollusca, one and the same thing, and arthropod or arthropods. Well, in these phyla, you can see that the mouth develops first and then develops ants. Okay. Now, talking about the deuterostomic animals, or we can say the deuterostomic evolutionary line. In this line, the animals they develop anus first, then develops mouth. So, anus is developed before mouth. Okay, and the examples to this line can be given as echinoderms and echinoderms and chordates. And when I say chordates, we also get into this group, vertebrates. So we are evolutionary. We are close to the echinoderms rather than annelids, mollusca, because in us, in we humans, we also develop ants first and mouth later. Okay? So this is the body plan. This also forms one of the important character for the classification of the kingdom of the animals. Now let's discuss coelom, which is an important character also, like others. So coelom. Now talking about the coelom, it can also be called as body cavity. We'll simply write it here as basic body cavity. Okay. Well, coelom is nothing but it's actually fluid-filled body cavity. Fluid-filled body cavity, and it's lined by the mesodermal epithelium on either side. Okay, and it lies actually between the body wall and the digestive tract, body wall, and the digestive tract of animals. Okay. All the internal organs are suspended in the coelom. Well, the coelom actually it has got many functions. It allows the greater body flexibility. Greater body flexibility. It also acts as a shock absorber. Okay, so for example, if a body gets a shock, it absorbs that shock and it protects the internal organs from the injury. Okay. Similarly, talking about the internal skeleton, it also acts as an internal skeleton. The best example is in case of earthworms. You can see earthworms. It makes their body turgid, and that is why it acts as an internal skeleton in earthworms. So, on the basis of the nature of the coelom, what is the nature of the coelom, and what's the nature of the coelom? We can divide the animals into three groups, and the first group is the acoelomates. When there is A, it means absent. So it means that the coelom is not present. Well, in other words, we can say the body cavity is not present. So we have here now that body wrote down that body cavity is absent. Okay, body cavity is absent, and the space, that space which should have been there but that space is not present, that coelom, that body cavity is not present, that space is occupied by the parenchyma-like cells. Okay, and the best example for the acoelomate group is the blood helmet. So in case of blood helmets, you will not find the coelom or body cavity. Similarly, talking about the pseudocoelomates, well, animals having these are those animals which are having false coelom. Okay, the body cavity is not formed from the mesodermal epithelium. Body cavity is not formed from the mesodermal epithelium. Recall that this cross represents negative. It means the body cavity is there, but it's not actually formed from the mesodermal epithelium. It should be lined on either side, from this side, from this side, from this side, from all the sides. It should be lined by the mesoderm. That is the middle layer. Okay, middle embryonic cell layer. Well, that's why it's called a pseudocoelomate, because it's not lined by the mesoderm on all sides. And instead, you can see that the mesoderm appears as scattered pouches in between ectoderm and endoderm. Okay, it appears as scattered pouches in between the ectoderm and the endoderm. Well, so now the point is, if this is a coelom, then how it is formed? How it is formed? It's actually formed from embryonic cavity called the blastocoelom. Okay, that embryonic cavity with the pseudocoelom rises. That's actually called the blastocoelom, and the ASCII element is the best example of this, because in ASCII elements you find out that there is pseudocoelom present. That's why the ASCII elements are called the pseudocoeloms. Now, coming to the third group, this was the second group. Coming to the third group, they are called the coelomates, or also called the eucoelomates. Now, these organisms, these animals, they possess true body cavity, or we can see the true coelom. Okay, so it means, what is that true coelom? It means that there is body cavity, there is coelom, and that is mesodermal in origin. It's totally mesodermal in origin. Okay, and what I mean to say, I will explain it, and we can say that on all sides, this body cavity is lined by the mesodermal epithelium, and this true coelom can be found in annelids, mollusca, arthropoda, Phynodermis, hemichordates, and chordates. Okay, so that's why they are called the coelomates, or we can say the eucoelomates or true coelomates. Well, I have here an image. You can understand from this image. See, this is the first image. This first image is the image of a coelom. Okay. Well, this is a platform. Okay, because we know that in platforms, you will not be able to find out there is coelom. Okay, there is no coelom. Well, this is the first layer, and this is called ectoderm. This is the second layer. This is called this red color layer. This is called the mesoderm, and this is the third layer, and green color layer, and this is called that internal, this white color. This is nothing. This is actually the digestive cavity. This can be found in all three layers. Okay, so well, there is no coelom. In the mesodermal region, you will find out that there is no coelom. There is no body cavity. That's why they are called the acoelomates. Well, this platform is an acoelomate. And the platform we are discussing here is here, and it's called the pseudobiceros bedpost. Okay, this is a species kind of platform. Okay. Well, talking about here, you can see the second image here. This is the image of eucoelomate. Okay, but before that, look at here. Look at pseudocoelomate. You will find out that this is the pattern of how and here you will see that first layer that represented by endoderm, ectoderm, and the second layer that's called the mesoderm. You will find out that reddish color layer, this is mesoderm, and the internal layer that's this green layer, and this is endoderm. Well, we see here another layer, this pinkish color layer. This is actually nothing. This is actually the pseudocele. Well, why we call it as a pseudocele? Because this is not totally lined on the either side. On the one side, it is lined by, on the outer side here, you can see that it's lined by the mesoderm, but on the inner side, it's not lined by the mesoderm. That's why we call it as it's the pseudocoelom. Okay, so this is the example of the that are actually the endoparasites, and they are found in our body, especially in children. While this round ball here we are discussing is the heterodera glycides. Okay, so this is the example of the pseudocoelom. This is a false coelom or the false body cavity because it's not lined by the mesoderm on the either side. It's lined by one side. Now talking about here, this is a pseudocoelom. Okay, eucoelom. Now, in this eucoelom, you will find out that this outer layer, this is again ectoderm. This inner layer, this reddish color layer, this is actually mesoderm, and this internal, this small circle, green circle, this is actually, and that white is the digestive cavity. Leaving that digestive cavity aside, let's discuss here. See here, you will find out that this color, this pinkish color here, this pinkish color. You can see my cursor here, or my pointer here. This pinkish color, this is actually the body cavity, and you will see this is here. This is here on its outside. It's covered by mesoderm, and also on the inner side is also covered by this mesoderm. So it is not, this body cavity is not in touch with the endoderm or the ectoderm. It's on either side, on all sides. It is lined by the mesoderm, or so we can say it's mesodermal in origin. So that's why this is called a eucoelom, and we can say this organism is eucoelomate, and this is found in the case of the annelids, echinoderms, chordates. So the annelid we are discussing here, this pattern here, and this is actually the glycera. So this is the this is the eucoelom. We also can call it as a coelom or coelomate animal, and this is the true coelom, and this is the pseudocoelom, and this is mesoderm. Hope you understand the difference between all these three. Okay. Now our second last the character which we are going to discuss is the segmentation. Okay. Well, talking about the segmentation as also the main character in the as the basis of the classification in case of an animal or the animal kingdom, the segmentation in this, for example, if you will take the example of some animals, their entire body is transversely divided into number of ring-like parts. Their entire body is divided into ring-like parts, and they are called actually segments. And this phenomenon, this is actually a phenomenon. This is actually the natural phenomenon. This phenomenon of segmentation is actually called metaphors. Okay, in case of biology, it has got a scientific separate name. This is actually segmentation is actually it is just a common, it is just a trivial name. But in case of science, we have got a separate name, a special name for this, and that's called the metamorphism. Well, this pattern is clearly seen in annelids. For example, we can see the phylum Annelida. And in case of Annelida, we have here earthworms. And in case of earthworms, I will show you through an example here. Wait a second. Yeah, this is an example here. This is the example of earthworm here. And this earthworm, you will see here that it has here these cutting-like structures, and these are actually segments, and this phenomenon is called the metamorphism. Okay, Well, this segment is a little bit larger as compared to segments, and that's why this portion is called collateral. Okay. Well, this can be this is a separate topic, but let me show here that you have to just understand what actually segments look like. So this is the segments. Okay. Now coming back to the topic. So these segments actually they help these earthworms in the process of locomotion. Okay, the segments they just get compressed and they also get uh, like rare functions, just like in compressions and rare functions, and due to which it moves. Okay, they get contracted and they get expanded, they get extended, and due to which the earthworm moves from one place to the other place and shows locomotion. So segmentation helps us in the locomotion of the earthworms. Similarly, not in case of the annelids, you can also find these segments in the phylum Arthropoda. And in case of phylum, you will see the millipedes and centipedes which we have in our homes. And those domestic millipedes or the centipedes, as well as those forest millipedes or centipedes, which are very much poisonous. And in them, these segments can have appendages attached to them, just like they have legs, they have antenna, they have mouth, and these legs they help them in locomotion, and in case of antenna, they help in their sensory functions. Okay, so antenna are there for sensory functions and mouth for feed. Okay, so this is about the arthropodas. This is the metabolism. This is the function of the segments in case of arthropodas. Well, talking about also the insects, other insects here, they the metabolic segments are grouped to form the body regions like head, like thorax, okay, like abdomen. So this is called cephalization. Okay, these segments get modified to head, thorax, and abdomen in case of insects. Well, let's move to the another main topic here, and, and that is notochord. Okay. Well, what actually is notochord? What actually is notochord? Notochord is nothing. It's just like it's tube-like structure, like this. It's just a tube-like structure. Okay. And, these are sheets. This is actually well, this notochord is uh, it's mesodermal in origin. It's mesodermally derived, rod-like structure. Okay, and it's present along the dorsal side of the and above the alimentary canal. Okay, this is alimentary canal. This is actually present on the dorsal side and above the alimentary. Okay, based on the presence and absence of the notochord, we have divided the animal kingdom into the two groups, chordates and non-chordates. So the chordates are those animals which possess notochord. Okay, and in at some stage in their life, which was notochord at some stage in their life. Just like, for example, we have your fishes, we have birds, we have mammals. They possess notochord at some stage, and this notochord is actually forms the basis for the axial skeleton. Okay, it's for the basis for the axial skeleton in the late life. Okay, well, the another group here is that's not. Well, the non-chordates simply, as the name signifies, that non-chordates are those animals which do not possess notochord in their at any stage of their life. Okay, and the best example for the non-chordates is from Porifera to well, the porphyras, this is not a new name, these are actual saponins. Okay, so this was all about that we discussed those characters which formed the basis for the classification of the animal kingdom. Okay, the entire animal kingdom has been divided on the basis of these 7 important characters. Okay, so I hope you enjoyed the topic and you got all the concepts clear um, which we discussed in this topic, and I hope there will be no difficulty in understanding the rest of the chapter. So, goodbye, have a nice day, have a nice time. So, keep in contact with me, and if you really enjoyed this topic, don't forget to uh, comment, don't forget to hit the like button, and don't forget to subscribe my channel. Thank you very much.